What's up, everybody? Today we're going to be talking about recombinant DNA technology. So we're going to discuss cloning, DNA libraries, PCR, uh, gene therapy, and genetic engineering. And there'll be various subtopics throughout each of these points that we'll be going over. Um, so cloning, it's the process of creating an exact genetic replica of an organism, cell, or DNA sequence. And there are three main types of cloning. Um, DNA cloning involves creating multiple copies of a specific DNA sequence. This is achieved through the use of restriction enzymes, DNA ligase, vectors. Um, so the restriction enzymes, they cut the DNA at specific recognition sites. The DNA ligase seals the cut ends and joins the DNA fragments together. The vectors, they're DNA molecules that carry the clone DNA sequence into host cells. Um, and then we have reproductive cloning. So reproductive cloning involves creating an exact genetic replica of an entire organism. And this is achieved through somatic cell nuclear transfer, or SCNT, and embryonic development. The nucleus of an adult cell is transferred into an egg cell, which is then stimulated to divide. The clone embryo develops into a fetus, which is then born. Therapeutic cloning involves creating cloned embryos to extract stem cells for medical research and potential therapeutic applications. So some of the mechanisms involved in cloning. Gene cloning involves the isolation and amplification of a specific gene or DNA sequence. Cell cloning involves the isolation and cultivation of a single cell, which then divides to form a colony of genetically identical cells. Um, Organism cloning involves the creation of an exact genetic replica of an entire organism. Um, some applications here are basic research. Cloning is used to study gene function, gene regulation, and gene expression. Gene therapy. Cloning is used to introduce healthy copies of a defective gene into cells. Regenerative medicine. Cloning is used to generate stem cells for tissue repair and regeneration. Um, Cloning is used to improve crop yields, disease resistance, and nutritional content. Um, so those are just probably some positive effects from cloning. There are, of course, many negative effects. Um, I think the biggest one is probably ethical concerns. Cloning raises concerns regarding the potential for human cloning, the use of embryos for research, and the ownership and control of controlled organisms. Like, imagine if somebody had a patent over your life. Like, it's borderline almost slavery, right? Uh, Cloning raises concerns regarding the potential for unintended consequences, such as genetic mutations or disruptions to ecosystems. And cloning raises the concern of regarding the need for regulatory frameworks to govern the use of cloning technology. So just another thing, imagine if the government controlled your life and they told you what you could and couldn't do, and more so than what they would normally do. Um, DNA libraries. So what is a DNA library? It's a collection of cloned DNA fragments that represent the entire genome of an organism. DNA libraries are essential tools in molecular biology and genetics, and they allow researchers to um, have this collection of data, and they can represent an entire genome. Um, and of course, they can use all this um, for diagnosing certain diseases, or discovering issues in many types of organisms, but especially human, let's say. So there are different types of DNA libraries. We have a genomic DNA library, a cDNA library. Um, a genomic DNA library is a collection of cloned DNA fragments that represent the entire genome of an organism. The cDNA library is a collection of cloned complementary DNA, so cDNA, and there are cDNA fragments that represent the expressed genes of an organism. Um, Construction of DNA libraries, so DNA isolation, DNA fragmentation, vector preparation, ligation, transformation. Um, all these are constructions of DNA libraries. And DNA isolation is DNA that is isolated from cells or tissues. DNA fragmentation, the isolated DNA is broken into smaller fragments using restriction enzymes. Vector preparation, a vector such as a plasmid or bacteriophage is prepared to carry the DNA fragments. Ligation, DNA fragments are inserted into the vector using DNA ligase. And transformation, we've discussed this before. The vector is introduced into host cells such as bacteria where the DNA fragments are replicated. Screening DNA libraries. So we have hybridization, which is a probe is labeled to hybridize with the cloned DNA fragments. Sequencing, the cloned DNA fragments are sequenced to determine their identity. Um, some applications in a real medical setting, I guess we could say. DNA libraries are used to identify and isolate specific genes. That's gene discovery. The libraries are used to study gene expression and regulation. DNA libraries are used to assemble genome sequences. Some advantages and limitations. So there's comprehensive representation. DNA libraries provide a comprehensive representation of an organism's genome. They enable a high throughput screening for specific genes or sequences. There is a limitation as far as how complex and difficult DNA libraries can be to construct. DNA libraries can be biased towards certain sequences or genes. So PCR, polymerase chain reaction. This was actually very useful during COVID. PCR tests were probably the strongest test that we had to test for COVID. But PCR is a laboratory technique used to amplify specific DNA sequences. This method has revolutionized molecular biology, genetics, and forensic science. The principle of PCR. So we have denaturation. The double-stranded DNA is heated to separate the strands. Annealing, primers are short DNA sequences, and the primers bind to the target DNA sequence. We have an extension. DNA polymerase synthesizes new DNA strands by adding nucleotides to the primers. There are different types of PCR. We have conventional PCR, which uses thermostable DNA polymerase to amplify DNA sequences. We have real-time PCR, or otherwise known as qPCR. Monitors the amplification process in real time using fluorescent probes. So kind of like FISH. I don't know if you guys remember that uh, tool, right? Reverse transcription PCR, RT-PCR amplifies RNA sequence by reverse transcribing them into DNA. Some applications of PCR, we have genetic testing, forensic analysis, gene expression analysis, diagnostic testing. So PCR is used to detect genetic disorders and mutations. Um, in criminology, PCR is used to analyze data for evidence. PCR is used to study gene expression and regulation. And as I mentioned just a while ago, PCR is used to detect infectious diseases and pathogens. Some advantages of PCR is high sensitivity. PCR can detect very small amounts of DNA. High specificity. PCR can amplify specific DNA sequences with high accuracy. And rapid results. PCR can provide results in a relatively short period. A few limitations of PCR is contamination risk. PCR reactions can be contaminated with extraneous DNA. We have inhibitors. PCR reactions can be inhibited by certain substances. And non-specific amplification. PCR reactions can amplify non-specific DNA sequences. Uh, so gene therapy. This is possibly my personal favorite topic to talk about. So gene therapy is a medical treatment that uses genes to prevent or treat diseases. It involves the introduction of healthy copies of a defective gene into cells to restore normal gene function. Um, there are several types of gene therapy. We have a germline gene therapy, which targets reproductive cells to introduce genetic changes that can be passed on to future generations. Somatic gene therapy targets non-reproductive cells to introduce genetic changes that will not be passed on to future generations. Um, Methods of gene therapy. We have a viral vector mediated gene therapy. It uses viruses to deliver healthy copies of a defective gene into cells. Non-viral vector-mediated gene therapy uses non-viral vectors such as liposomes or nanoparticles to deliver healthy copies of a defective gene into cells. We have gene editing, which uses gene editing technologies such as CRISPR or Cas9 to directly edit the defective gene. Some applications in gene therapy. So genetic disorders, cancer, infectious diseases. Gene therapy can be used to treat genetic disorders such as sickle cell anemia and cystic fibrosis. Gene therapy can be used to treat cancer by introducing genes that inhibit tumor growth or promote tumor cell death. I believe I think just this morning in Korea, they were able to reverse cancer cells to become healthy cells again. So a major breakthrough on the other side of the world. Infectious diseases. Gene therapy can be used to treat infectious diseases such as HIV by introducing genes that inhibit viral replication. Some advantages. Gene therapy has the potential to cure genetic diseases by correcting the underlying genetic defect. Long-term treatment. Gene therapy can provide long-term treatment by introducing healthy copies of a defective gene into cells. Um, Reduce risk of side effects. Gene therapy can reduce the risk of side effects associated with traditional treatments. There are many limitations as of modern day, as of today, I guess. Gene therapy can have safety concerns such as the risk of off-target effects or the introduction of new genetic mutations. Gene therapy can have efficacy concerns such as the difficulty of delivering healthy copies of the defective gene into cells. Cost concerns is probably the biggest. Gene therapy is extremely expensive, and it is mostly inaccessible to, or it is inaccessible to most patients, pretty much most patients in the world. Um, but I've mentioned this before. I think gene therapy is the future of medicine. I think it's the key to eradicating all disease and any disease. Um, so I think as time goes on, it'll definitely get better. The research will get better. The cost will eventually decrease, or even if it increases, I think it'll be to at a point where, I mean, the success rate is 100%, and it's totally worth, um, you know, whatever amount it may cost. I guess that's the question I'd leave you with is, what is your health worth? What's the price for your health? Um, so we have genetic engineering. which is also known as genetic modification. It is direct. It is the direct manipulation of an organism's genes to introduce desired traits. This is achieved through the use of biotechnology tools such as recombinant DNA technology and gene editing. So, types of genetic engineering. We have transgenic engineering, which involves the introduction of genes from one species into the genome of another species. We have cisgenic engineering, which is the opposite. It involves the introduction of genes from the same species or a closely related species. Gene editing involves the direct editing of an organism's genome using technologies such as CRISPR or Cas9. Some methods of genetic engineering. We have recombinant DNA technology, which involves the creation of recombinant DNA molecules by combining DNA from different sources, gene cloning, and gene editing. Some applications of genetic engineering. We have in agriculture, genetic engineering is used to develop crops with improved nutritional content, increased resistance to pests and diseases, and enhanced tolerance to environmental stresses. In pharmaceuticals, we have genetic engineering being used to produce therapeutic proteins such as insulin and growth hormone. Biotechnology, genetic engineering is used to develop new biofuels, bioproducts, and biomaterials. So advantages, in agriculture, it improves crops yields and reduces the needs for pesticides and herbicides. It's used to produce new therapeutic treatments and proteins, therapeutic proteins, that is. Genetic engineering can be used to develop, as we said, biofuels and more sustainable fuels than traditional alternatives. There are some safety concerns. Genetic engineering raises safety concerns, such as the potential for unintended consequences or the introduction of new allergens, or even the creation of a new bioorganism, that we could even say. Regulatory frameworks. Genetic engineering is subject to regulatory frameworks that can vary by country and region. Yeah, which raises the issue again, like in countries that don't have any regulation, you could have an outbreak on your hands. And I mean, we've seen it before, just 5 years ago, right? Genetic engineering can be subject to public perception and acceptance issues, much like cloning. Sorry about that. I had a technical difficulty there. So genotype and phenotype. The genotype is the complete set of genes that an individual organism possesses. The phenotype is the physical and behavioral characteristics of an organism that results from its interaction of its genotype and the environment. So I've discussed Mendel before, but his experiments on pea plants demonstrated the fundamental principles of inheritance, including the laws of segregation and independent assortment. Mono and dihybrid crosses. A monohybrid cross is a cross between two organisms that differ in a single gene or trait. A dihybrid cross between two organisms that differ, or it is a cross between two organisms that differ in two genes or traits. Neo-Darwinian synthesis, also known as modern evolutionary synthesis, integrates genetics, evolution, and other fields to explain the diversity of life on Earth. The statistical analysis of genetic data using the chi-square test. It is a statistical method used to analyze genetic data and determine whether observed frequencies differ significantly from expected frequencies. Statistical analysis of genetic data, and the objective is to analyze genetic data using the chi-square test and determine whether observed frequencies differ significantly from expected frequencies. So some materials that you'll need to conduct a chi-square test is a calculator, a genetic data set, and the chi-squared table. So in order to complete this, you review the genetic data set and identify the observed frequencies. You calculated the expected frequencies using the null hypothesis, and you calculate the chi-squared statistic using the observed and expected frequencies. Determine the degrees of freedom and look up the critical value in the chi-squared test table. Compare the calculated chi-squared statistic to the critical value and determine whether the observed frequencies differ significantly from the expected frequencies. And so that's all I have for you guys today. I hope you guys enjoyed the video. I hope you guys learned something. Again, if you have any questions, you could always ask me, or you could always ask whatever research or tier 1 research a tool you have at your disposal. AI has improved significantly here in recent years, so even asking AI would be a great way to learn. But I hope you guys are well. Hope you guys are safe. I hope you guys enjoy time with your family and your loved ones. And I'll see you guys in the next video.